Hi everyone. Hello and welcome to our course, The Systems Biology of Aging, Aging, Aging here and there. Aging, 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 aging everywhere. Aging is a song that is universally sung. Aging is a thing you can't ignore if you are young. But when you are older and begin to have dementia, you'll think back upon this course and wish you'd paid attention. Aging, aging, aging here and there. Aging, 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 aging everywhere. Aging, 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 aging everywhere. Aging. So as we go into the unknown together, it's always good to have a ritual that we all know, like the theme song, right? So let's all take a nice deep sigh of relief. Such a privilege to do some science together. So now it's lecture 9. We're going to talk about genetics of aging and longevity. And in our course we had a kind of arc of the course. Coming close already, we can already see the end. You know that there's a final project that we put the instructions on the website. It's basically to open your creativity and choose a topic that you find interesting in a broad sense about aging and answer it with the approaches and concepts we studied in the course. The approximate length is like one of the questions in the exercises as a guideline. And it's good to do it in pairs. Any questions about that? Yeah.
If we have something we want to do that's not necessarily age related, but time related or something like that.
Yeah, so you can talk with Ben and get approved. So in our arc of our course, we started with some of the patterns of aging, and we talked about the facts, right? The hallmarks of aging, the molecular hallmarks of aging, and then we talked about mathematical model set with the houses and trucks, and about diseases of old age, and then we talked about exercise and decline, how that fits in, nutrition. Maybe it's a circle. Nutrition and longevity. We talked about the weight set point, remember? Yeah, and then we talked about animals, right? Comparative longevity, immortal animals. Comparative longevity. And what we can learn from immortal animals, and also very long-lived animals like whales or bats that are long-lived compared to their mass. They have a high longevity quotient. And we asked what superpowers they have in terms of their cells, and we saw that they have really good DNA repair, especially double-stranded DNA repair. And that a lot of their accuracy increases when you compare species. The longer lived they are, the more accurate they are in different processes, like translating proteins, and errors in their chromatin positioning, things like that. A mutation rate, maybe a cancer defense mechanism. So even non-dividing cells in humans, like neurons, reach 3,000 mutations at age 60, and mice reach the same at age 2. And mutations rise more or less linearly with age because of chemical processes, not necessarily. Divisions add another few mutations per division. So rapidly dividing tissues like colon, lining, they have a little bit more mutations, but it's mostly time passing that gives mutations. And mutations are not the clock for aging, because we know that human beings born with defective DNA polymerase that makes much more mutations, they don't age faster. They get more cancer. So I used to think maybe mutations rising linearly with age are the houses, but that's not the case. I mean, They are houses, and maybe they'll kill you if you live to 1,000, but they're not the one to kill you first. So today I want to talk about what is it that kills you first. And what are the first two things that kill you first? We know the first two things, probably. And then you guys will have to discover the third. Yeah.
Didn't we say that the houses are stem cells that accumulate mutations?
We said that the houses are stem cells, and they don't accumulate mutations, they accumulate epigenetic modifications, which are not this. I mean, of course, they accumulate mutations, but the relevant houses, we think, today I'll tell you why, are specific changes in histone acetylation. Yeah. And so today we're going to do the big reveal of what we think is the best guess for the molecular clock, very specifically. That's good for you, Alex, and Neta, because you're going to measure it. So why histone 4K16 acetylation? So now we're going to talk about the genetics. And today what we'll do, we'll learn from yeast, because there you have good genetics. Then we'll skip across this tree of life to humans, and we'll talk about diseases of rapid aging. So what mutations cause rapid aging? And then we'll go to centenarians, people who live to 100, and they have enough lung capacity to go. Half of them have no diseases at age 100. I mean, none of the top 10 diseases of age at age 100. There are only 3 out of 10,000 of the population. But what genes what's their genetic secret? Is it the opposite of the defects that give you accelerated aging? How does it relate to the long-lived animals? So we'll talk about that. The genetics of aging. And then what's in the future, you might ask? So I want to cover social, cognitive, and emotional aspects of aging. The cutting edge of the biotech, of the drugs that can extend health span and maybe lifespan. So where the field is right now. And I wanted to have a nice goodbye party with all the songs. Any questions? Yeah.
Will we talk about sleep?
Oh, sleep. You have a request to talk about sleep?
Yeah.
Okay. Let's put it here. I did talk about sleep, do you remember? At the beginning of last lecture? Were you here?
Yeah.
So I talked about adenosine and the circadian. I'll talk about sleep. There's something. It really ties very nicely to Alzheimer. Why do you ask?
Because you were talking about 4 pillars.
Oh, the 4 pillars, the 4 legs of the elephant of health:
Like
nutrition, exercise, sleep, and social relationships. So let's talk about sleep too. Good idea. Okay. This is an interactive course, guided by your questions. Any more questions or comments about the arc of this course? What do you think? Yeah. What do you want to? What are you curious about? Which of these topics is most curious for you?
maybe sleep.
Sleep.
Yeah.
Okay. We have a double vote for sleep. That's great. Okay. To sleep. Perchance to dream. Yeah.
If you could say anything interested to us about what are the problems or the ideas, like what are the things that you think that you might wrong or what you want to understand
So what are open questions in the field?
Yeah.
Great. I can do that. Yeah, where we're at it, yeah.
You talk about extending lifespan. Will you talk about reversing the aging?
Can we reverse aging? So what are like the most advanced thoughts people have about that topic? What else? This is great. This is like your menu. Yeah.
Maybe also like how different organs might age different rates.
So I can write here clocks and organ clocks. Yeah. What else? This is great. I might have to add another lecture. Yeah.
Stress.
Huh?
Stress.
Yeah. So it's emotional is like anti-chronic stress. So what's the connection between chronic stress and the biology of aging? We happen to be experts in that. Yeah. Yeah, but what is interesting for you?
Well, I actually was curious about stress.
Stress, yeah, great. What else?
Mitochondria.
Mitochondria. We'll talk about that
Cycle
today.
nature.
Huh?
Connection with nature.
Connection with
Aside from
nature,
alleviating
yeah.
stress.
What is it about connection with nature that seems to help with chronic stress?
And sleep and wish.
What do you think?
Vastness, like the ability to step out of your own problems in your own life. And
Yeah,
like
so
fix-
if you're in a setting that's vast, like I'm standing at the cliff of the big crater in the Negev, and the scenery is vast, it creates a cognitive vastness that helps me maybe be a metaphor for taking better perspective on my problems.
it's one of the ways.
More in touch with the immortal Atman inside me, which is like nature, always there and perfect just the way it is. Yeah. That's a nice challenge. I never thought about it, but it could be nice. What else? Yeah.
Maybe about evolution and aging.
Mm-hmm. So I try to talk about that in the animal, but you want to know more about evolution, okay?
Yeah,
Like
they're
what?
trying to defeat evolution.
Huh?
All of the drugs and trying to make us live longer. It's also like trying to defeat the evolution.
Okay. So is that so? So we're trying to defeat evolution. What do you think about that?
I don't know.
Can we? There's also this question: why study aging? I mean, is it ethical to study aging? Yeah,
Also maybe the microbiome and aging.
microbiome and aging. Okay. What else? Yeah.
Is there something to the concept of the blue zones, or some cultures have it figured out?
Yeah, I'll talk about it maybe today with the centenarian. Yeah.
What is it built out?
Yeah, I'll tell you. Yeah.
How aging affects the immune system.
Okay. Okay. Good. Aging and genetics, they go hand in hand. Aging and genetics, they go hand in hand. Aging and genetics, they go hand in hand. There's a clock, I'll help you understand. Helping and genetics, they go hand in hand. You know, some people live for 100 years. Some people live for 100 years. They smoke their cigarettes and drink their beers, and yet they live for 100 years. How can they smoke and still not choke? 'Cause genetics and aging, they go hand in hand. You know, genetics and aging, they go hand in hand. Genetics and aging, they go hand in hand. There's a secret clock, I'll help you understand. 'Cause genetics and aging, they go hand in hand. We'll learn from yeast, the magical beast, how genetics and aging, they go hand in hand. All right. So we'll start today with yeast. Why? Because almost everything we know about the cell and about the longevity genes comes from very simple organisms, actually. A lot of it from worms and a lot of it from yeast. Then they're verified in flies. Then they're tested in mice. And then compared to human studies, etc. But for some reason, the yeast, which is a cell quite similar to ours and it has a nucleus, it's a eukaryotic cell, unlike bacteria, is when you ask what genes make it live longer, you get genes that are conserved all the way to humans. So that's amazing, even though we're separated by a billion years of evolution. There's something about the basic machinery of the cell is conserved, really doesn't change fundamentally somehow. Of course, humans have immune systems, hormones, fat, muscle, brains. The yeast doesn't have. But on the cellular level, there's a lot of on the single cell level, there's a lot of similarity. So that's why it's useful to study yeast. They're also they smell good. They're like bread. Also, so how do you study aging in yeast? There's two ways. One is called chronological aging. You starve them, like I told you about E. coli. They all die different times. You ask which, if you delete a gene, do they live longer? But that turns out to be less informative about human aging. There is another kind of aging that is much more informative. It's called replicative aging. Replicative aging you count how many buds or daughter cells a mother can make. So the budding yeast, Saccharomyces cerevisiae, it doesn't divide like this, it buds. And then this bud becomes its own cell. Yeah, that's called budding yeast, right? This is a non-sexual reproduction. It also has a sexual reproduction, in which case it's more like the fertilized egg, makes a spore. Spore flies, lands on a grape. The grape is very, very toxic to bacteria. So it's like this spore landed on a planet. It's the only one. It's better than bacteria in having all these checkpoints. So it's very, very careful because it has one shot. And then it makes the bud. And then the bud makes another bud, and the mother makes another bud. So now the two become 4. The 4 become 8. But you can still keep track of that original mother cell. How do you do that? You grow the cell on an agar plate, and every couple of hours when it divides, you take a pipette and you dissect away the bud. This is a hard experiment, right? Because you have to sit there. And then you get tired, you put it in the fridge. You come back tomorrow. And it turns out that the wild type yeast can do about 20. And you can plot the survival curve, generations. By the way, generations work well more than time because the last generations, the cell cycle becomes longer and longer, and generation is better for our purposes and for all the field, and this is survival. So 100% of the mothers survive, and then it goes down like this. And the T50 is about on the order of 20 generations. It's a long experiment. Now, the way you look for genes, another reason to use genes is the awesome power of yeast genetics. So, till recently, it wasn't possible in human and mouse cells to do what you could do in yeast. Now it's possible. You can read in the DNA, write in the DNA, whatever you want, very easily in yeast, because you can put in DNA and recombines into the genome. It's very elegant. You can, for example, early on, scientists developed a library of all deletion strains in yeast. So each strain has a different gene deleted. So there's about 4,500 genes that yeast can survive out of the 6,000 when you delete them. And you can do double deletions. It's very, very... So suppose you delete a gene, So now you don't have a certain gene, make some protein, and you're looking for things that extend the lifespan, right? And then you say that gene is a longevity gene. And then you have the mother and the bud, and yeah.
Then the bud move apart, or you cut it while it's still there?
Yeah, so it buds, and then you need to remove it.
Okay.
So that otherwise it'll grow exponentially. I mean, it'll bud too, and you'll lose track.
That doesn't know what happens to the child.
Yeah, you take it away. Okay, so this is like a, let's call it longevity deletion mutant. You can also overexpress, which means you put the gene under some strong promoter and you make more of it of the protein product than usual. So you can, and when you look at so when you have thousands of genes and there's a few hundred that can really change the lifespan by a lot. But what's by a lot? I think like if you do a screen, the maximum the single gene can do is about 40% extension. If it was a human being, that would mean 40 years. That's a lot, but all right. So and there's all the hallmarks of aging I talked to you about that are cellular come up here: DNA repair, proteins, mitochondrial genes. But like I said, I told you in the lecture number 2, the field is now at the place where there's so many things that can extend lifespan, and they're so interacting that there's not a sense of something, let's say, more important than the rest that drives them. But that's where we can come in and use our math model to find a candidate mechanism to drive aging. That's what I want to do with you. So, for example, there's this heroic study in yeast by Kennedy, McCormick, Caberlin, who taught for years. They taught a yeast genetics course, and they had undergraduates do this experiment on thousands of deletions. Every year, a whole class like this, each person gets a gene, does this experiment. They amassed data on more than 2 million budding events in this way, and made a data set, and looked for families of genes that extend longevity. And they have a very nice paper in 2015. This paper gives you the half-lives. So you say this gene, deleting it goes from 20 to, let's say, 24. And what we would like to do now together is to use not only the T50 but also the entire shape of the survival curve. Why? In order to understand if this gene deletion is in the houses, damage production, in the trucks, damage removal, in the threshold, or in the noise, so that we can have better resolution to understand the aging pathways. So by now, we talked enough about this model. You understand that it explains a lot with a little, so let's apply it to this problem. So our goal is to find the Holy Grail. What is the first molecular events that are like in the houses that drive this thing? And then we'll look, are they also in humans? Is there a case that they're also in humans? It's very important. It's a high moment in the course for me. And it was really fun to develop, because by learning how to teach it, I also understood more about what we discovered. This is work Tomer here, Levi. Can you say hi? And also Navera has also contributed something very important. So how can we do that? So when you look at the survival curve shape of the yeast, I'm going to transform it here to hazard, which is risk of death per generation. It rises. But instead of a Gompertz law like in humans, it actually looks more like this. It goes quite quickly to a power law. That was weird. It has an exponential part, but around gen 10. So if I analyzed how the SR model what are the parameters that give you this shape, and came to a shocking discovery, and that is that this is indicating that the yeast has no trucks. Whoa. So he says no trucks. It's just houses being built linearly, and they make damage, and it crosses the threshold. And noise saves you somehow. Okay, and just to say that houses, so if you have dx dt equals eta t, x goes like 1/2 eta t squared, which you might remember from high school. If you have a velocity, this is like acceleration, so this is, and this reaches x critical at around 20 generations. So it's perfect, and the noise would reach x critical if it was alone in about 140 generations, which means the noise is kind of smaller than the acceleration, but still plays a role. That's the parameters in yeast. So actually it's a simpler problem. We don't even need to worry about the trucks. We just need to find what the houses are, what the threshold is, and we have a better chance to understand what the garbage is. That's what we want. What is the garbage? And what are the houses? Okay. And what's going to be our tool? So our tool is going to be a graph like this, where here it's the T50, how long it lives, and here is the steepness. So we're going to look at survival curves, look at their T50, and also their steepness, how steep they are. Okay. So you can define it in different ways. Our favorite way right now is to look at the distribution of death times, and look at the coefficient, the standard deviation, and look at the mean divided by standard deviation. So 1 over the CV. So it's like how broad this is. It's very simple. But you can define it also as the interquartile range divided by the median. Doesn't really matter. Did I answer your question? That is the steepness. Okay. And it turns out that if I change eta, if I change the houses, I'm supposed to be on this line. So more houses, I'm short-lived and steep in this case, and there's no trucks. And in the other, if there are trucks, you get scaling, but short-lived. And if I change the threshold, I'm short-lived and shallow. So this is shallow. So if I have a mutant that's short-lived and steep, I can say it's in the houses, or it's making an age-dependent source of damage. And if it's short-lived and shallow, I can say it's in the threshold. So what do you mean by the threshold? So I have here the houses making garbage. There's no trucks. This is the threshold. If there's another process, let's say this is going to be the houses that look like this, that's making a garbage that's also competing on the same threshold, but it's at steady state. So let's call this x prime. I can say that this effectively lowers the threshold. So if I make garbage damage, the same kind of garbage, not in an age-dependent way, steady state, and both of those need to cross the threshold, it's like effectively lowering the threshold. The yeast is more fragile because it has this extra damage. So if I delete something that makes the same kind of garbage, but it's not age-dependent, it helps me understand. So I'm going to say that, and each mutant I can plot here. I take the mutant, I look at the survival curve from a corner. By the way, they send us kindly their whole data set so we could do that. Otherwise, can't do that. And of course, there's some error bars because there's a few tens of you. So let's say the mutant falls here. We say, oh, this looks like probably in the houses. Or maybe it's long-lived and shallow. Oh, probably in the houses. And oh, here's a mutant that falls here. Oh, it's probably threshold. And there's also noise. I don't know anything. I can't explain anything about the noise. So I'm just going to draw. This is what happens when you change the noise. Okay, so you. But I don't know what to say about the noise, as usual.
The protein in the middle is survival.
What do you mean in the middle? This thing?
Yeah.
Yeah, survival. Thank you. This is lifespan distribution. This is the cumulative distribution of this. Is like the chance of living more than. And here we take this. We analyze the median and the steepness, and we plot each survival curve. Now is a dot here. And from where it falls, we can estimate which parameter it changes. Did I explain myself?
What was the result?
Huh?
What was the result?
Yeah, I'll tell you what the result is. But before that, I want to make sure you understand the method. I explained the method.
Can you explain again for like one specific gene? What do we do?
Yeah. Let's suppose, Tomo, what's your favorite gene?
You can go for the APPs.
First of all, most of the genes, they don't affect the survival curve. And so they fall here. This point is the wild type curve. So most of them fall here, or here, or here. They're not different from the median and steepness of the wild type curve. Most. I mean, a few hundred are different. A few thousand are not different. Let's take one that's different. For example, in the yeast, there's a vacuole, which is like a big garbage collecting thing. It has a lot of other functions. And here's a pump that pumps proton in. And if you delete that pump, you get something that's short-lived and shallow, short-lived and steep, short-lived and shallow. So it'll look like something short-lived and shallow. It looks something like this. This is the deletion strain. Okay, so now I have the wild type. So each experiment, of course, you do the wild type and the deletion. And now I look, what is the median? The median is lower than the wild type. What is its steepness? Its steepness is lower. You see, it's less shallow. So it's actually here. That's one mutant. And now I can do that for 1,000 mutants.
And why do you say that it was related to the threshold?
Because I know the model, and now in the computer I change X critical, and I get everything. When I change X critical, it falls on this line. When I change houses, it falls on this line. Of course, both parameters can change at the same time. Then it'll be somewhere in the middle. But it's safe to assume that a deletion would usually affect one, not two. Sometimes you have two. Okay. Did I explain myself? Great. So this question is, I'm sure other people had the same question. So that's our methodology here. You can apply it, of course, to any organism or, except that if there are trucks, the houses will be on the x-axis because they're scaled, they don't change the steepness. In yeast, you have this. Okay. Now what are the results? Just kidding. So this is genetics. Genetics is a very interesting field in biology because it takes you on a trip, because the genes can be anything. You scan all the genes. They can take you to any part of biology. It's so beautiful. To work together with Dan Jarosh, Tomer Levy. Other people contributed, Avi, Mayo, Ifan, and Young. If the screen isn't working, you get all kinds of genes you can't make sense of. In this case, you get very coherent sets of genes that kind of tell a story. So now it's an interpretation act. right? So when you look at the houses, what you find is genes that acetylate and deacetylate histones. You know? So as you remember, DNA is a string. In the humans, you can have a meter into a 1 micron nucleus. That's because it's wrapped around these proteins called histones, and also in yeast. And these histones bind the DNA. They're generally positive charged, and the DNA is negatively charged. When you acetylate, that is to say, add a chemical group to certain lysines, it neutralizes their positive charge, and they unbind. And then what happens is that regions which are closed and hidden suddenly become open. And they can be transcribed, they can be read, they can be damaged. Right? So what you find, the houses are things that write and things that erase specific out of the many possible acetylations. Lysine number 16 on histone subunit 4 and histone number 56, lysine number 56 on histone number 3. So it's so specific. Are going to be the first events that we can detect. Okay. Also in the houses, we know it has to be in the telomere region, which is the end of the DNA, because in the houses we also find complex of proteins that protects the end of the DNA. Otherwise the cell would think it's a double-stranded break. And so in the telomere region, telomeres are normally very, very closed chromatin, because there's no genes there to transcribe. But what these things do is open it. And this increases with age. And the reason it increases with age, people might be because when you do have double-stranded breaks there and you try to repair it, and you manage to repair it, that shifts the histone acetylations. And you're not meant to particularly care about that because it's a rare event. But if you're a mother cell and you keep the bad DNA, the mother cell, that's going to accumulate. Those changes changes that open the chromatin. This is something that increases with time. And indeed, we know that this H4K16 acetylation in yeast and also in humans rises with age. This one decreases with age. But either increase or decrease of this one shortens life. So it could be that both of them are bad. This one, okay. So that's something in the telomere region, the end of the DNA, about the chromatin structure. And those particular residues are increasing. So it's like increasing acetylation. So for example, if you knock out the eraser, you have more acetylation, and therefore you have shorter lifespan, and in this case, steeper curve, and you know it's a house. So you kind of split up the houses. Yeah.
These then all kind of damaged together, or you look at the weakest link?
I'm just telling you now which genes come out.
Yes, but when you look at both telomere ending and like it sounds like there are different damage that can kill you differently.
Yeah. So far, this doesn't kill you.
Okay.
This is something that now the interpretation of the genetics.
Yeah.
One interpretation, right? We have to interpret is that the driver of aging is specifically those acetylations in the telomeric region that open it up. Okay, and then what kills you?
Yeah.
So what kills you? So the next thing you find from this analysis is now it's open. The DNA is open, and we know we get this RNA polymerase. So we have RNA is copied, and in places they're not supposed to be transcribed. When you transcribe them anyway, you get something called R loop, which is RNA DNA hybrid that shouldn't be there, and also G4 quadruplex, all kind of structures that shouldn't be there. And how do we know that? Because we find all these enzymes that cure R loops. They're like X critical. So if you don't have them, you have R loops all over, and that contributes age-independent garbage that lowers the threshold. That's how we figure out that R loops are important. And you know that R loops will happen there because of open DNA and stuff like that. What's wrong with them is when the DNA tries to duplicate, you have this DNA polymerase and it tries to duplicate, and when it collides with problems like that, you get double-stranded breaks. So I'm sorry for the people who don't know biology. It's like a very elaborate mechanism, but you get, the genes you find are sitting on this pathway. So you get, for instance, these helicases, like, I don't know, 8 genes that are responsible for clearing the way for DNA polymerase. They're called the GCM complex. You get all the double-stranded break repair, especially the ones in the telomeres. But you don't get other kinds of repair, like single letter repair, base excision repair. You get very specific kind of genes. And so this is, you would say that if you want to look at the molecular clock for this, you need to look at the telomeric region and ask how much acetylation there are in histone 3 and 4. That's exactly what Alex and Netta are doing, or only Alex, in Liran's Shlush's lab now, on human blood. So we're jumping all the way to human, right? We'll talk about how this relates to human. This RNA helicase in yeast, remember, because it's going to show up in a human disease that causes accelerated aging. So it's like connecting. But that's not all. It turns out yeast, like an onion, has 2 kinds of garbage. How do we know that? Because when we starve the yeast, caloric restriction, it lives longer. And there's a whole set of mutations that no longer affect longevity. These ones do, but there's other ones that don't, and that's how we can separate it into 2 different pathways. And so what's the other pathway? What's the other layer of the onion? It has to do with mitochondria. Somebody asked about mitochondria. So this is again going to be a kind of a specific damage pathway. I don't think you could think about without doing this. It's like very specific. So the mother cell has these fibers that help it to keep damaged things inside the mother cell. That's well known. So the daughter cell is fresh. So what does it keep? It keeps many things. It keeps some kinds of proteins, but it keeps the vacuole, which is, I told you, quite a big, it's also called lysosome in human cells. It's quite a big acidic vacuole where you put in damaged proteins and it breaks it down to amino acids. It also stores amino acids, it stores protons. It's good for homeostatic of pH and amino acids and stuff like that. And so it keeps the old vacuole and also keeps the old mitochondria. And mitochondria are also tethered to the vacuole. And sends the fresh mitochondria and fresh vacuole to the bud. What do I mean by fresh? So it becomes, with age, the vacuole becomes less acidic, which is bad. It needs to be acidic. This is fresh, it's acidic. And the mitochondria have less membrane potential, the old ones. So the mother cell becomes, every generation, it keeps more damaged stuff. That's the way it's designed. It's designed is good because otherwise we wouldn't have an immortal cell, right? It's like, it's for the daughters. The daughters are fresh. The daughters of old mothers have the same longevity. They're reset, except if the mother is very, very, very old. It's so sweet. Okay, maybe it's the father cell. I don't know. Maybe mother cell. Fathers also keep the damage. Keep the damage. All right. So what's going on with this? So what we find is like Tomer's favorite gene. You find, for instance, all the 10, 20 subunits of the proton pump that keeps the vacuole acidic. If you delete them, find them. And in the mitochondria, you find, again, it's very, very specific. So it turns out, I'll tell you the story. It's not that this less acidic means it doesn't degrade proteins well enough. That's not the problem. The problem is it stores amino acids, and when it's not acidic enough, there's too much amino acids in the cytoplasm. And which one? Cysteine, this one amino acid, which is kind of toxic. And this cysteine chelates iron, and when there's not enough iron, you don't have iron-sulfur clusters to carry out the electron transport chain, which is the way to make ATP in the mitochondria. And specifically, complex 1 and complex 2, and quinine. So what I'm telling you here is so detailed because you know which deletion is houses and which deletion is threshold, and you can put a line in the middle of the 4 complexes of electron transport chain, and you know that then they leak reactive oxygen species O2 minus into the mitochondrial matrix, causing all kind of terrible things, protein damage, DNA damage. So you can tell this very specific mechanism. And you can say, why doesn't yeast evolve a way to make their vacuole acidic again? So it does. But that has a certain capacity, and it's not designed to be old. So by generation 4 or 5, by generation 5, you're only 2 to the minus 5, one of 32 cells. By generation 10, you're one of 1,000 cells. By generation 20, one of 1,000,000 cells. So there's not selective pressure. It's like, who cares about that? So we have, and by the way, I want to say that also in human neurons, vacuoles become less acidic. That's one of the first things that happens before Alzheimer. Vacuoles become less acidic, then mitochondria are damaged, then there's protein aggregates, plaques, and stuff. That's in Alzheimer, Parkinson, and Lewy body dementia. In all of them, you have this biomarker of aging. This is 2 kinds of death. Okay, death by mitochondria and death by double-stranded break. And it turns out that there's actually findings in yeast that correspond really well to that. That's by Nan Hao and UCSD, who, by the way, solved this problem of having to wait and dissect by making microfluidic devices that catch a mother cell and then it buds in a flow. And with a computer, you can count. And they discovered yeast has 2 ways of dying. So some individual cells, genetically identical, make round buds, and other ones make elongated buds. These ones have mitochondrial problems. These ones have some kind of DNA problem. They couldn't find out what it was exactly. And yeast, in a very early generation, 1 or 2, decides how it's going to die. It has 2 repair systems that are in a toggle switch. And it flips. Either you repair mitochondria and you die from DNA, or you repair DNA and you die from mitochondria. And then they did the following experiment. Oh yeah, by the way, if you only express one of the 2 systems, you die by the other one. That's how they can separate these 2 modes of death. And it turns out that the DNA one is shorter lived generation than the mitochondrial one. So they said, okay, let's express both systems at once. And they got genes that live a little longer, but it wasn't that great. And then in another paper, they did one of the most beautiful experiments in the field. This is a physicist called Jeff Hasty that I know from my system biology days. He's an expert. So what you said, when you express both of them at once, why is there a toggle switch? Why does the yeast need to decide one or the other? Maybe their functions collide somehow. You can't do both at the same time as well as each one individually. So they said, let's separate them in time. So what he did was design in the cell a synthetic or artificial oscillator that makes HAP part of the time, SIR part of the time. And Jeff Hasty is an expert in that. How do you do that? That's the field of synthetic biology. You change the logic. Instead of a toggle switch, you turn it into a negative feedback loop. And this circuit oscillates. This circuit is either this one or this one. If you just change one arrow here, you get a circuit that's an oscillator. So one goes up, activating its inhibitor. Okay. And it worked, and you got oscillations that are longer than a cell cycle. And when they did that, they got the longest living yeast. Did I do something wrong?
The loop on the cell can stabilize.
Oh yeah, right. You're right. They got rid of that.
Okay.
Thanks. So wow, these guys? So some people here, have looked at oscillators. So when you oscillate like that, this is like published I think last year, you get the longest lived yeast ever from a genetic mutation, 82% extension of lifespan. So I thought that was pretty. Oh, and the yeast die with good vacuoles and good mitochondria and good DNA from something yet unknown. So there's a third layer to the onion. That's your job to find out what it is, okay? So it's an onion. I want you now to ask you to parent share, find a biologist and ask your question about something here that's less clear. Or if you're two biologists, just discuss it, okay? So don't leave anyone alone. Maybe it's someone behind you, Baraket, and talk about it. Okay. Any questions that arose that I'd love to hear? Yeah.
What is the distribution of all the genes on this graph?
What is the distribution of all the genes? So it's in your lecture notes. The distribution of all the genes on this graph. So this is the longevity, and this is the steepness. So most of them are here, and then there's something like this. something like that. So there's lots of more. They're more X critical, but there's definitely a lot of ETA genes. So a lot of ways maybe to make the yeast more fragile, and a few that are we interpret as having affecting the time dependent aspect, which is most interesting for us. I mean, everything is interesting. Yeah.
Do you have an explanation why, for example, if the mitochondria damage starts, why it eventually leads to protein aggregation and like
Yeah, I do.
next
So
stages?
right. So mitochondria is like, imagine it's like a furnace burning the sugar, and in that reaction there's lots of it's oxidizing it, and there's lots of chance for oxygen to not connect and become superoxide, which is very reactive, and that reacts with proteins, DNA membranes, and there's enzymes in the cell that they're really like fire fighters, the like SOD1, SOD2, which are huge X critical genes. Like, all right. So it's easy to see that in this case, these two enzymes, they leak O2 into the matrix of the mitochondria, and they're inhibited by this lock of iron, because that blocks basically the iron sulfur cluster here. So right here is where the electrons can't move anymore. So if you delete ubiquinone, you get it. If you delete complex 1, if you delete complex 1. So this superoxides are going to destroy proteins and make them aggregate the mitochondrial DNA, the membranes. Then they're going to leak out and destroy everything. So they're very bad. And of course, you also don't have enough ATP. So you actually see the cell cycle start to really become long in the old mothers that die in this mode, because there's not enough energy. So both of those things will kill you. Answer your question?
Yes.
But it's this particular ROS, because there's other kinds of reactive oxygen species. So we have a lot of things to test here. And of course, this connects to vast literature that you can connect and understand. Other questions? Yeah.
How can, like you said, exercise somehow, I don't know anything about biology, but exercise somehow affects mitochondria, I don't.
Yeah, it does.
So how does it have to do with genetics? How does it contradict it?
Okay, so I talked about exercise. So one of the positive things that exercise does, anaerobic exercise, the mitochondria are where you take oxygen. So it has the lowest oxygen concentration because it's where oxygen is used up to burn. And if you do anaerobic exercise, the mitochondria sees, I don't have enough oxygen, I need more mitochondria. So you build up mitochondrial capacity. The more capacity you have, the less prone you are to damage. You can say. So that's one example, but many other. You actually build up everything, your breath, your heart, your blood vessels, and your mitochondria to pump more oxygen and get it to make more energy. So that's a connection. And mitochondria damage is especially bad in neurodegenerative diseases and also some kind of cardiac diseases. And we'll talk about that after the break. How do we jump to humans? Because you just jump to human, right? From yeast to human, which is so very natural. That's what we're going to do too. And yeah.
We basically have the two kinds of damages that are independent.
Yeah.
So do we have two independent thresholds? And if we try to like put two
Yeah.
mutations, what happens?
So we do have to. Yeah. So the question is, there's two damages. So we need two SR models with two sets of parameters and two thresholds.
And it works.
Yeah. If you let the cell repair both kinds of damage, it dies by something else. You can genetically make it only die from one or the other by upregulating the other, and then you can see that everything fits. So, for instance, even caloric restriction, it basically abrogates the mitochondrial path. So caloric restriction actually, this was actually discovered by Nan Hao. Remember, there's two modes, DNA and mitochondria. If you do caloric restriction, they only die from mitochondria. Is that right? No, it's the opposite.
Show me.
What's faster?
The mitochondria die first.
Yeah, sorry, I confused. I apologize. Mitochondria die before the DNA. But if you do caloric restriction, they don't die from mitochondria, they only die from the DNA, and that's why they're longer lived. And that's what we also see when we look at dietary restriction. A lot of things that were houses now are no longer doing anything, and that's how you can separate genetically the two pathways. Or if you delete SIRT2 or HAP4 and do a double mutant, you can also separate it out. So if you upregulate one repair system, some of the houses are no longer houses. So that's how you can tell there's two families. That's genetics. And it's okay. Now it's 3. Now we want to take a break. Yes, I was asked some questions in the intermission that I thought were fascinating. So I just want to say that this particular lecture, preparing it, was really part of research. It was really helped us consolidate what we're thinking. And now these questions also surprised me with some things we never thought about. So I really thank you guys for participating. So I was confusing you, but it's the mitochondria pathway. So early on, the yeast upregulates either mitochondria or mitochondria biogenesis, regeneration, antioxidant resilience, or high histone deacetylation. Therefore, abrogating one of the problems. And for some reason, it doesn't do it both of them at the same time. This is a kind of bet hedging, it's called in biology, where in the first generation, you landed on the grape, you don't know what's going to happen. So you say half of the cells are going to be good for mitochondria, half of the cells are going to be good for DNA, not for aging, but when you're young, and that's called bed hedging. That's a lot of microorganisms do that. One or the other repair. Now, it was asked, what about exercise, right? And reactive oxygen species, how does exercise help out here? So reactive oxygen species used to be the dominant theory of aging by Harman and all that. And then it turned out that they actually have a good side. Low levels of reactive oxygen species are a signal for the cell, like the HIF system, to upregulate antioxidants. It's like hormesis. You stress the cell, and what doesn't kill you makes you stronger. So if you do anaerobic exercise, it's weird, but you're training your mitochondria. They become less fragmented. There's more mitochondria, so you can make more ATP. So there's enough oxygen. If there's not enough oxygen, they're full of enzymes that can deal with reactive oxygen species, like SOD1, which is a matrix antioxidant, SOD2, or the other way. So your mitochondria become better, more resilient, able to work at low oxygen, and give you the energy you need when you're exercising. Why does exercise help Alzheimer? So the neurons, I told you, are especially as if they die from this mitochondrial mode. The vacuole, the mitochondria. So it's like you're also training your brain. It's very interesting. So the reactive oxygen species at low levels is a good signal, makes you stronger. But what I'm talking about here is a major disruption. So it's massive oxygen that outstrips the SOD1, SOD2 ability and destroys it. In our cells, if mitochondria become breached, mitochondria, remember, are bacteria-like things. So when you have exposed your mitochondria to the cell, you're also turning on immune response to bacteria. So even more problems. So it's like in our cells, it's very bad if mitochondria become compromised. So this doesn't mean DNA damage, because reactive oxygen species make DNA damage. When I say DNA, I mean specifically that histone 4K16 acetylation, this pathway, the telomere shortening, double-stranded break. That's what we think is going on here. And there was also a question that said that NAD links these two, so maybe it's a big link. And that's known to be a big factor in aging too. For Tomo, that's for you to find out, because I didn't think about it, but mitochondria make NAD, and that's the thing that's missing to cause this oxidative damage. And SIRT2 uses up NAD to deacetylate. So, great comment. Thank you for that. Anyway, moving on. I'm going to jump now from yeast to our centenarians, okay, to humans. So how does this relate to humans? the genetics of longevity in humans. And to do that, I'm going to erase. So fun to teach. It's like part of research when it's such a young field where everything is open and the stakes are so high. But if you're wrong, maybe an equation dies, no patient dies. So, unlike like doctors or something. All right, and so the circle is not closed, right? We have a lot of things to discover where you can fit in. Also, if you're watching us at home, you can do it. Come join us. Actually, my vision is that lots of people aren't working on aging, like working on making a better cell phone, would come and study aging or other aspects that are closer to human well-being. Okay. So I'm going to talk about two things in humans. One is called diseases of accelerated aging. These are exceedingly rare, thank God. The defining one is called progeria for aging, gerontology. It's called Hutchinson-Gilford progeria syndrome. And it's caused by a specific mutation in a gene that speeds up aging by a factor of 7. So what do I mean by that? That you can see it already in the very young toddlers. Start making a lot of phenotypes that are like human aging. White hair, brittle skin, muscle problems, cataracts, hearing loss. They die of heart disease and stroke in teenage. Terrible. So you see a lot of phenotypes of aging, not just one, not just cancer or just Alzheimer. A lot of them. Now, this mutation is so rare. It's not you don't inherit it from your parents. It's a somatic mutation. That is to say, it happens after your parents determined your DNA, and it happens like in 1 in a million, something like that. They're very, very rare. It's dominant, so only one copy needs to be mutated. And what this mutated lamin protein does is, this is the cell. This is the nucleus. The nucleus has this very important lamina, which is like a container, and that's where the DNA telomeres are sitting and are silenced. And this lamin A mutation makes the nuclei irregular and unsilences or opens up the telomeres. Do you notice the connection? So that's the progeria disease. Now, I'm really amazed by how few, and I'm grateful, how few diseases there are that speed up aging. Because if there was many ways to speed up aging, we would see many. So, there's thousands of genetic disorders. You're born with mutation, unfortunately, that's in your mitochondria. You get, let's say, a lot of lactate and metabolic problems, but you don't age. You're born with mutations in your I don't know what. Some essential stuff, if you don't die in embryo, most of those mutations kill you. But if they're like mild enough, you get specific things. But there are very few ones that accelerate aging in many, many phenotypes, okay? It's exactly evolution. Now, this is there's a few others that are not exactly progeria, they're progeroid, which means they share a lot of symptoms. It's not as much as progeria. And there's one that's most close. It's called Werner syndrome. It's a mutation in a gene. And here you die in your 40s. But again, it's like, let's say, accelerates aging by a factor of 2. Now, what is this mutation? Here, you need to get mutations from your mother and from your father together. It's in REQ helicase. So it's this one. The yeast analog is SGS2, whatever is there. So that's the second one. And then there's a few others, and they're all DNA repair. There's Bloom, which is also an analog. Cocaine syndrome, which is a transcriptome-coupled nucleotide excision repair that works here. And that's about it. I would say 4 syndromes. And how do I know that? Because there's no beta, X-critical DNA, mito, 2 ways, progeroid syndrome. Because Morten Schiebe Knudsen, who's an MD in Denmark, who's a brilliant aging researcher, did a bioinformatics study. He took a database of symptoms of diseases. So each disease is a vector of symptoms, like gray hair, cataract, blah, blah, blah. And he did clustering to see which diseases have shared symptoms. And before that, he defined the symptom of healthy human aging. And I put that in the lecture notes. What is healthy human aging? What happens with like hearing loss, loss of hair? And he find, reassuringly, that these diseases are the most similar to aging. And that's it. And the next most closest are defects in DNA repair that don't give you accelerated aging, but give you some. The next most closest are mitochondria diseases, but they're quite different. So it's something about this kind of DNA repair, very specific, that can accelerate aging. I find that it's like a genetic experiment, very unfortunate in human beings. So it must be very specific drivers of aging, and also that hitting them is usually lethal. You don't even see the baby born. So I don't know how to figure it out, but that's what I can tell you. Now, another very important fact for me is that these diseases, they spare the brain. What does that mean? You get cancer, diabetes, heart disease, but what don't you get? You don't get the 4th horseman. Isn't that interesting? That, here just going to speculate, that human beings also have 2 ways to die. We have the stem cell, differentiated cells, senescent cells, inflammation, all the aging-related diseases, cardiovascular, cancer. They have more cancer. And what else? Diabetes, right? Because these are dividing cells. This acetylation pathway gives rise to double-stranded breaks, blah. In the non-dividing, so this is in the body, in the brain and other non-dividing cells, I bet you it's the vacuole, mitochondria, ROS. Why? Because we know that in the major neurodegenerative diseases, Alzheimer, Parkinson, Lewy body dementia, first step, vacuole deacidification. Second step, mitochondrial damage. Third step, protein aggregates, either amyloid plaques or synuclein plaques, tau, etc. And that's why these DNA repair, which come up in the histone acetylation pathway, don't affect the brain. Did I explain myself? And there are separate mutations that give you early Alzheimer. Maybe we'll talk about them. And they don't affect your body too much. There's, of course, they do, but not like progeria. That's what I want to say about progeroid diseases. Any questions before I go to centenarians?
You mentioned some of the hallmarks of these aging is like, let's say, white hair or baldness. Like, so let's say you have someone who like has like baldness when they're 30 compared to someone who's not bald when they're 30. Does that mean that the other person who's bald is like aging at an accelerated pace, or?
Okay, here's the question. I said alopecia, hair loss, is a hallmark of aging. If a person loses hair at age 30 rather than at age 55, does that mean they have accelerated aging? So if they have one of those diseases, they do, but you'll see it in everything. But in general, remember each disease has an XD. So hair loss in men and women is different. In men, it has to do with the derivative of testosterone. And the threshold for that is genetically determined. So you can be susceptible or not susceptible at young age. So that's a separate thing. What you die of is separate. But eventually, most men get some kind of hair loss. Not everyone. Cataract is almost universal, I would say. Hearing loss, there are more. Did I answer your question? So each disease has its XD. Each phenotype has its XD that can be genetically determined. So, hair loss, of course, is probably not a good marker for aging clock. So interesting. But maybe if you connect a lot of phenotypes, that is a good aging clock, and that's exactly what's called the frailty index. Not exactly, and that is a good marker for aging. So if you collect a lot of things, you can overcome the genetic differences. Right?
Specifically says it don't include hair.
What?
It specifically includes graying hair.
It does include. It does include.
Specifically says that will not.
Oh, it says don't include.
It might.
In frailty index, people are careful not to include gray hair because that's so genetic. Right? Yeah. Anything else not to include? Anything that doesn't really correlate with age. I mean, gray hair does correlate with age, but not as well, right? Or hair loss.
That's something everybody can get.
Everybody can get. Okay, good. So this is Glenn Pridham, who's a postdoc from Canada, working on aging. All right. So let's talk about centenarians. Happy birthday to ya. So centenarians, there's a question. Yeah. No, let me continue. I don't have enough time. Ask me later. Okay. The questions are very enriching for me, but I also. So centenarians are about 3 in 10,000 people make it nowadays. The number of centenarians in the world is growing like this. So there's going to be a lot more data, a lot more birthdays. There are some of them that are called delayers. They get age-related diseases, but a few decades later than not most people. Some of them are called avoiders. They don't get aging-related diseases. So about half of them get to 100 without heart disease, metabolic disease, or cancer, except for benign skin cancer that everybody gets, BCC. Wow, what's their secret? So understandably, researchers, since there were centenarians, which is about 1920s, starting to look at their genetics and their lifestyle. So first of all, the lifestyle, that's the blue zone, which is, by the way, non-scientific, but there's a wonderful Netflix series I really recommend, the blue zones. The places where there are lots of centenarians, like Okinawa, like Sardinia, like Southern California, a religious order, like Costa Rica, the Nicoya Peninsula, the great places to visit. People say it's more correlated with poor places where there's forged birth certificates. But that doesn't matter because they have such a wonderful lifestyle. Like in Sardinia, they have these villages where they're steep like this, so you have to climb all the time. And in Okinawa, they love their old people, so they get the social relatedness, and they're like, just venerate them. And in Nicoya Peninsula, it's Costa Rica. What need I say more? A country without an army. So it's amazing, right? So it's nice to watch, but But the point is that when you look at those centenarians, like Nir Barzilai collected Ashkenazi Jew centenarians. Why Ashkenazi Jew? Because you want them to be more genetically related so that you can look at the difference between centenarians and non-centenarians without worrying if one is from one ethnic group from another, right? Want to control for that. So he says, look, I come to this one, I say, what's your secret? And he says, drinking 3 whiskeys a day and smoking a cigar. So basically, they have something that can make them protected from things that would kill the rest of us pretty quickly. It's not that they have lifestyle secrets, except that maybe there's a slight bias towards optimistic point of view, but maybe that's because they outlived all their enemies and their doctors and their lawyers. But of course, they don't have nobody. They don't know anyone who remembers what it was like to be in Berlin 1905. Right, that's sad. But that's it. Apparently more optimistic than the average. And when you look at their genetics, you find recurring genes that occur again and again in different populations, like in Ashkenazi Jews, in South Italian centenarians, in Chinese Han centenarians, and some that are more specific to each group. And you can think, maybe it's the opposite of progeria. Maybe those genes are genes that give you better DNA repair. And the answer is no. It's not that. So that's also interesting. There's 2 kinds of genes. There's X-critical genes and there's ETA genes. So the big class is protective gene variants. So unlike mutations they're very rare. These variants are differences in DNA that are very common, like more than 1% or something like that. So you and me, we have 3 billion letters, and we're different in about 1 of 10,000 letters. We're different. We have a million differences. And some of those differences are quite common. They appear in 1% population, 2%, 5%, 30%. And those variants come up, right? And so they protect you against diseases. Like there's ApoE2 that protects against Alzheimer and metabolic disease and cardiovascular disease. That's a big one. They don't have ApoE4. So this one protects, this one activates. This has to do with cholesterol. They have CETP, that has to do with cholesterol. They have some nicotinic brain receptor that makes you addicted to smoking. You have a variant that makes you less addicted to smoking. Those are the shared things, right? And they have, they have Klotho, which is a gene that even if you have ApoE4, you won't get Alzheimer. It's a protective gene. In other words, they're a delta hand with protective genes and without the disease-causing genes of the 4 horsemen. Okay? That's it. So that's one class. Oh, and also reduced inflammation. So IL-6 variants, HLA, DQ, which is MHC class 2 variant that gives you autoimmune diseases. All kind of genes I wrote over there that are variants that are anti-inflammatory. That's good because in our model here, senescent cells cause inflammation, right? So these genes anti-inflammatory, they work here on the X-critical region. So each centenarian has like one of them or 2 of them, I don't know. It's heterogeneous. And there's many different variants that give the same effect, you can say. And then the other class of genes is in a pathway, again, that we heard about in this lecture at least 3 times, is cell. So this is protective gene variants against the horsemen. and there's cell resilience, which is, I would interpret as House's gene. And that is, again and again and again, hitting the insulin-like growth factor 1 mTOR pathway and the transcription factor FOXO3, which is a transcription factor inhibited by IGF-1. So if you lower IGF-1, its activity rises, and it does DNA repair, lots of good stuff, a lot of repair. Now, these genes are also found in worms. So DAF2, DAF16. These are like the first aging mutations in yeast found. This is the homologs of these genes. By the way, all these resilient genes are found also in worms, flies, mice. So they're very conserved. I don't know why not others, but it's this pathway. And I told you before, this pathway is thought to tune the cell. So if it's starving, repair and recycle. If it's not starving, you can grow. IGF-1 hormone tells cells to grow. If there's enough nutrients, as you know, from insulin and amino acids, mTOR, you grow. The body reproduces, etc. And if you downregulate that, you're more resilient. So those centenarians have lower circulating levels of IGF-1. They also have higher HDL cholesterol and lower LDL cholesterol, this is the bad cholesterol, this is the good cholesterol. That's in this area. Here they have IGF-1. Anyway, that's what you can learn from so far from centenarians. I think this is going to improve a lot because there's much more centenarians now, and there's improved mathematical models because you have a person has 1 million variants. So you're doing a statistical test between centenarian and non-centenarian on 1 million hypotheses. So you need multiple hypothesis correcting errors, which are very, very strict. So there's also analytical methods improving to fish out genes more. Again, a very growing field, so we'll probably find more. But in the context of our model that we're building here, you can see that you can talk about EDA because of this repair. The cells are more resilient here. Or protecting from the stink of the garbage, the inflammation, or the consequences. Diseases that can protect against a lot of horsemen at the same time. What do you find in centenarians? Yeah, there's no beta, you know? That's weird. Yeah, why not more NK cells or something? Why are there more centenarians? No, it's more centenarians because a centenarian now needs to be born in 1924. But centenarians 20 years ago were born in 1900. So there were much fewer people in 1900 than 1924. You know, there was 1 billion people, now we're 10 billion people. It's growing like this. And extrinsic mortality has gone down. So there's more centenarians now than ever. Japan, for example. It's like, you can walk down the street and see a centenarian. Super centenarians, 110. Japanese women. Semi-super centenarians, 105. Some people live to 100 years. Some people live to 100 years. They smoke their cigars and they drink their beers. Some people live to 100 years. How can they smoke and still not choke? Genetics and aging, they go hand in hand. All right. Thank you for all your questions. Let's take a nice deep sigh of relief.